protein synthesis, which is basically the making of proteins. So the word synthesis is just the posh way of saying making something. So protein synthesis is the making of proteins. Now this is what I would consider one of the hardest topics in the GCSE biology course, but by the end of this video you should be able to fully grasp what protein synthesis is. So we can split protein synthesis, so the process of making proteins, into two main stages. We have stage 1, which is called transcription, and we have stage 2, which is called translation. So you could imagine transcription and translation as two different things. So first, this is a diagram of a DNA, right? Transcription is basically the shortening, so making this DNA as small and simple as possible, taking just the parts that are needed. That's transcription. So making this as small as possible. And then translation is taking this smaller version of DNA and translating it into amino acids. Now, in case you didn't know, amino acids are what makes up proteins. So these different amino acids combine together in order to make one protein. So the process of making proteins, so protein synthesis, is you take a DNA, you make it smaller through transcription, and then you translate that smaller version into amino acids so that they can collectively join together in order to form a protein. Now that's just a short, simple example. Short, simple definition, I'd say. Let's dive into it a lot more deeper now. So, transcription is the process of taking a single gene of DNA and copying it into a structure called an messenger RNA, mRNA. So, a single gene is what codes for one protein. So this is a DNA, right? If we make this into a 2D diagram, we're going to get something like this. Now this is still a gene, it's just we flatten things out in order to make it a bit more simpler. Now genes are small sections of the DNA that code for a specific sequence of amino acids. Now when these different sequences of amino acids combine together, that's how proteins are formed. So from this huge and long DNA structure, we can take a small portion of it. So this DNA might be from here up to here. And then from here onwards, it might be another DNA coding for a different amino acid, forming a different protein. So this might form one protein. And then this might form for another. So we take this part and then we make it into a 2D diagram. And DNA has what's called a double helix structure. You've probably heard of this thing before. A double helix structure. I can't spell suddenly. A double helix structure. It basically means it's just a spiraling double-stranded structure. So if you look at this DNA diagram, it has two different strands. It's two different strands that are joined together through these colorful things called bases. So bases are basically the steps of the ladder. If you look at this as a ladder, the bases are basically like the steps joining these two different strands together. So DNA has a double helix structure. This is really important. You do need to know this. Now from here, we're going to have strand 1 and strand 2, right? And in between them, we have bases. Now bases, this is really important. You need to know these bases. So we have adenine, thymine, cytosine, and guanine. So for DNA bases, we have 4 different bases: adenine, thymine, cytosine, and guanine. So these are the 4 bases that makes up the steps of the double helix structure. Now adenine, thymine, cytosine, and guanine, this is quite complicated to remember, right? So there's a pretty simple acronym to remember. So it's apple tree in the car garage. Apple tree, adenine, thymine, car garage, cytosine, and guanine. Now the order of this is quite important as well. The order between AT and CG doesn't matter, but the order of AT, like essentially these two and these two stick together. The apple tree makes sense, right? The apple's on the tree, so they stick together. And car garage, the car goes in the garage, right? So these two would stick together. Now why is this important? It's because these bases, right? They bond in specific orders. So for example, if we had an A here and a T, if we had an A here and a C here, it would have to be apple tree car garage. In the same way, if we had a G and a C here, it would have to be car garage and car garage, right? So car garage always bonds together an apple tree and TA, so this would also work like that, TA. It always has to bond in the order of apple tree and cytosine guanine. So ATCG. It can't be TC or AG together. It has to be A and T and C and G. The order between these two doesn't matter. So it could be T and A or A or T, but it just has to always be AT and CG. So apple in the tree, car in the garage, adenine, thymine, cytosine, guanine. You do really need to know this. So these are the bases that connects the two different strands of DNA together, right? So we need to make the DNA strand much smaller. Now, why do we have to do this? That would be quite important, right? We need to know why this process needs to occur first before we can find out what transcription and translation is. So why do we need to do this in the first place? Well, I'm going to draw a rough diagram of a cell here. A cell, right? And most cells have their DNA in their nucleus, including us humans. So our DNA is contained in what is called the nucleus, the brains of the cell. Now, if you know basic cellular structure, you'll know that the DNA, the cell, is surrounded by lots of ribosomes. So these are ribosomes. And you should know by this point that ribosomes synthesize proteins. So are you making a bit of a link there? Synthesize, right? Remember, synthesize means make proteins. When protein synthesis is the making of proteins, right? Protein synthesis, making of proteins. The ribosomes synthesize proteins, so the ribosomes are what actually makes the proteins. So protein synthesis is actually looking at how proteins are made by taking the genes from the nucleus and then taking that to the ribosomes for the proteins to be synthesized. Now we have an issue. This is the ribosomes, right? Ribosomes are located within what is called the cytoplasm. Now the issue is the DNA is in the nucleus, and the nucleus and the ribosomes are not connected together, right? So the DNA has to exit the nucleus, and it has to make its way through the cytoplasm into the ribosome. Now the issue is that DNA, right? I'll try and draw a rough diagram, is incredibly long. It's way too big to fit out of this nucleus and to go into the ribosome. It's way too big for that. So what transcription, the process of transcription, right? The process of transcription is basically just making DNA smaller so that it can fit into the ribosomes, right? So it can exit the nucleus and so that it can fit into the ribosomes. So to use a gene, we need to make a copy of it first in order to make it a lot more smaller so that it can leave the nucleus. That is transcription. Why is it smaller? So we have a DNA, right? If we go back to the previous page, we have DNA, but only parts of it, called genes, are needed to form different proteins. The entire thing doesn't form one protein, it forms many different proteins. So only a section of it is needed for one protein. This will be another process of mRNA. Another process of mRNA will occur here, and this will be another process of mRNA. The entire DNA isn't needed. So only a single gene needs to be copied rather than the whole DNA, which is way, way, way much larger. Therefore, a copy of just the single gene would be small enough to exit the nucleus, and it'd be enough to make the protein that is needed and make its way into the ribosome. So the copy of this gene that codes for a specific protein is called a mRNA. In long form, it's called a messenger RNA. So you could imagine it as a messenger RNA because it's taking the DNA, becoming smaller so that it can be messaged. It can be sent to the ribosomes to be formed into a protein, right? So it's sort of acting like a messenger, getting smaller so that it can be sent. So there's quite a few differences between the messenger RNA and the DNA. And you do need to know this. So the differences between the DNA and the mRNA, which is the smaller version so that it can be sent to the ribosome, right? So let's take a look at the differences between mRNA and DNA. Remember, the mRNA is the shortened version that we made from the DNA, consisting of just a gene that's needed to form just a single protein. So, first of all, this is pretty obvious. It's basically the main concept that you need to understand. It's much smaller, right? Because it only contains one gene. It's only a single gene long, because only a gene is needed to form a protein. Number two, it's only a single strand. It is single-stranded. So compared to a DNA, which was double-stranded, right? It has a double helix structure with two strands. mRNA only has a single strand. We'll explain why in just a moment when we dive into transcription. It's single-stranded. We'll look at this in just a moment. And rather than containing the base thymine, so we looked at ATCG, right? Apple tree, car, garage. Rather than containing the base ATCG, it contains uracil instead of thymine, so AUCG. We'll look at this as well in just a moment. These are the three key things that you need to know. It's much smaller than DNA. It's single-stranded. It's much smaller because it's only one gene long, because that's how long it needs to be to form a protein. Single-stranded, and it doesn't contain thymine, and it contains uracil instead, so AUCG. Now let's take a look at the process of transcription. Okay, now this is when it starts to get a bit more challenging. But if you follow along, I think you'll be able to understand what essentially happens. So by the way, I just got these diagrams from the Cognito YouTube channel. Check them out. They do good explanations. But I wanted to dive into this a lot deeper so that you could fully understand it. That's why I'm making a bit of a longer video than the Cognito videos that are already available. But by the way, thank you for these diagrams. They're fantastic. So this is a gene, right? And if we make a gene into a 2D diagram, we're going to get something like this. These are called the bases. Remember? A, T, C, G. Apple, tree, car, garage. Adenine, thymine, cytosine, and guanine. Right? Now, this is not an mRNA yet. It's not a messenger RNA. It's just a gene that's been put into its 2D form. So we could call this an uncoiled diagram. And remember, DNA this isn't a gene. I miswrote. It's not a gene. This is a DNA. And we're going to get the gene from the DNA. And it has a double helix structure, double-stranded. So we have strand 1 here, and we have strand 2 here, 2 strands. And the uncoiled diagram is this 2D version. And these bases here are what connects the 2 different strands together. Now this might be way too slow for you, but going slow really helps you to consolidate what we've just talked about till now. So this is the uncoiled diagram, and this is the gene that we want to copy into mRNA form so that it can be smaller, so that it can exit the nucleus and go into the ribosome, right? Now we have something called RNA polymerase. Now remember, this is the gene that we want to copy. So this is the gene that's going to code for a protein. So 3 of these would code for amino acids that would then form a protein. So this is quite important. This is called a RNA polymerase. It's an enzyme. And if you remember what enzymes are, they're basically biological catalysts that speed up reactions. So we have RNA polymerase. It's an enzyme. Now this is really important. The RNA polymerase performs a really important function. So this RNA polymerase binds to the DNA. Remember, this entire thing is called the DNA. It binds to the DNA right before the gene being copied starts. So this is the gene that we want to copy, and this is the only section that we need in order to form the protein that we want to form, right? So it's going to bind to the DNA right before where it starts. Okay? Very important. Right before where this gene starts that we want to copy into mRNA, the RNA polymerase binds to the DNA. And what it does is it causes the DNA strands, right? 2 strands. It causes it to separate. So before, when the base is ATCG, right? Adenine, thymine, cytosine, and guanine, right? They would have been connected together. They would have been holding the 2 strands together. But this RNA polymerase bonds just before the gene that we want to copy. And it creates this, it unzips it. Imagine this as a zipper. it kind of unzips the things holding it together. It unzips and it separates these bases up. Now, as these get separated, it's going to form a space in between for the mRNA to form. Now, this is where the mRNA actually forms. So, the RNA polymerase binds to the DNA just before where the gene being copied into mRNA starts, right? It binds just before where this starts. And it unzips the gene, and it like unzips the two strands off so that we can have a gap in between the two. So just ahead of the DNA RNA polymerase, the two strands of DNA separate, like a zipper being unzipped, and this causes the bases to be exposed. A, T, C, G, adenine, thymine, cytosine, and guanine, they're going to be exposed now. And just like the DNA bases, our mRNA bases are also complementary to the DNA bases. So what happens now is, now that it's been unzipped, it's going to form the mRNA. So the blue, this is the mRNA. Now this diagram looks incredibly confusing, I know. So I'm going to explain this in detail, just a second. I'll first explain with this diagram and then I'll draw my own in just a second. So we have the DNA and it's been unzipped. As it gets unzipped, now this bottom thing is called the template strand. Now a template is what you use in order to create something new, right? So we have two strands of DNA. We can't use both to make mRNA because mRNA, remember, if you remember the differences, it is single-stranded. It only has one strand. So it only needs one strand because it's going to be complementary to the DNA, right? So it only has one strand. So it only needs one strand to make a copy from. So it's going to be the template strand, and essentially this other strand, I think I'll just do this in red to differentiate a little bit better. This other strand is ignored. We don't need to look at this other strand. Now the template strand is the only one that we look at, and it's the one that we make the mRNA copy of. So if we look now, we have Okay, you know what, this is just going to get confusing. Let me just draw in here. We have the RNA polymerase that's unzipping the DNA, and we have the exposed bases. Remember, this was the template strand. CTAGC. Let me copy that down here. CTAGC. Now, what does C bond with? Car garage, right? So this is going to have the mRNA bonding of G, guanine. T, what does T bond with? Apple tree, right? So it's going to be adenine. AT, right? Apple tree. But remember, mRNA has no thymine. DNA has thymine, but mRNA, also including tRNA, has no thymine. We'll look at tRNA in just a second. Essentially, mRNA has no thymine. There is no thymine base in mRNA. Instead of thymine, it has a base called uracil. So this is the DNA base. In mRNA, we have AUCG. Now the uracil bonds with the adenine in the same way, but the T is just replaced with the uracil. That's the only difference. So this A, right? So instead of a T with the mRNA, we're going to have a uracil there. G is going to bond with C as usual, and C is going to bond with G as usual. So we're going to get the mRNA of GAUCG. And if we go to the next diagram, you can see that we have GAUCG being made. That is what transcription is. It's where you take the base strand, so the template strand that you're going to make an mRNA copy of, and you take the template strand, and then you find the complementary mRNA base for that template strand. And remember, there is no thymine in mRNA, so it's always going to be uracil instead of thymine, where the thymine would have gone if it was DNA. That is essentially what transcription is. You combine the template strand with the mRNA complementary strand. These are called complementary because they complement each other. That's essentially what transcription is. It's when we form the mRNA. And this is what we call the mRNA. And remember, it's single-stranded. So this is the thing that's going to get sent off out of the nucleus into the ribosome to be synthesized into proteins. So we're going to get this. It's going to separate. The important thing about this RNA polymerase is it separates the DNA strands right in front of it, and it closes it just behind it, so that only a small section of the DNA is ever exposed, right? Now that the important thing, yeah, 2 strands of DNA separate just ahead of the RNA polymerase and close just behind it once it passes. This ensures only a small portion of the DNA will ever be exposed. And once the RNA polymerase finishes making an mRNA copy of the gene it wants to copy, so it took the template strand and finishes making it, right? Oops, that's the next page. So once it finishes it, it detaches. This RNA is going to detach, and it's going to close back the DNA up into its original form. So once it's done making its mRNA copy, it's going to close, it's going to leave, and it's going to leave it in its original closed form. So now we're going to be left with an mRNA strand that is free to leave the nucleus to the ribosome to make proteins. So we have the mRNA single-stranded, remember, single-stranded mRNA strand that will now be free to make proteins. So we have the nucleus, DNA, and we have the single-stranded mRNA strand. So for example, AUCG, I don't know, A, and it's the single-stranded mRNA strand that's free to leave the nucleus and go to the ribosome to synthesize proteins. All right, now let's take a look at translation. So transcription was the taking of DNA and making it smaller so that it can exit the nucleus. Translation is the taking of the mRNA and making proteins with it, essentially. Wow, this video is really long. My voice is starting to hurt. But anyways, we now have the mRNA. The blue is the mRNA strand. Remember, it's single-stranded. Now, the mRNA and ribosome bind together alongside tRNA to form a protein. Now, this is translation, okay? tRNA stands for transfer RNA. Transfer RNA. So we had the what's it called? mRNA, right? A messenger RNA, and exited the nucleus, and now it's binded with the ribosome. This is what's called the ribosome. It's what synthesizes proteins in order to make them, right? Ribosome and the mRNA has now binded together with the ribosome, and these are what we call tRNA molecules. Transfer RNA. And on top of them, we have amino acids. We have 1 amino acids in 1 tRNA. Now, tRNA and mRNA, these, believe it or not, these also form complementary base pairs. I'll just end that complement. Base pairs. These also form complementary base pairs. So adenine is going to bond with uracil, guanine, cytosine, uracil, adenine, cytosine, guanine. And remember, tRNA has the same properties as mRNA. So it has no thymine. And it has uracil instead of thymine. So when we have the tRNA and mRNA forming complementary base pairs, there's going to be no thymine to be found. Thymine is only found in the DNA. So these also form complementary base pairs. Remember, it's A, T, C, G for DNA. And because we don't have a thymine and we have uracil instead, so adenine and uracil will bond together with mRNA and tRNA, and cytosine and guanine will bond together here. Now, the transfer RNA has the same bases as mRNA, and that's how they form complementary bases, right? They also have adenine, uracil, cytosine, and guanine, and they form complementary base pairs. So 1 tRNA molecule, this is 1 tRNA molecule, this is 1, this is 2. They have 3 bases in it. 3 bases. 1 tRNA has 3 bases, and they bond with 3 mRNA bases. So that's what we call the bonding of these 3 bases. It's kind of like a triplet, right? And yeah. So we're going to get amino acids that come on the tRNA. And let me just check what my next page is. Okay, my next page is useless. So we have tRNA bases. Let me just get rid of all these drawings. It's kind of getting distracting at this point. So we have these tRNA bases, right? tRNA. And this is also another tRNA molecule. It has 3 bases in it, and this bonds with 3 mRNA bases. Now, 3 of these, like so, is called a codon. Or you could also call it a triplet. So as the tRNA and mRNA bind together in the ribosome, they form a chain of amino acids, right? So there's going to be another tRNA with 3 bases joining here, forming a complementary bond, another one, and so on and so forth. And they're all going to have another amino acids. And this is going to form a chain of amino acids. For every 3 mRNA bases, the ribosome lines up one complementary molecule of tRNA, each with also 3 bases. And the mRNA and tRNA form a complementary strand. So we call every 3 bases a codon. These 3 things are called a codon. Finally, these amino acids are made every time the codon meets with a tRNA molecule, right? So every 3 of these, triplet, meets with one tRNA molecule that also has 3 bases, and this is going to form one amino acid, one amino acid, one amino acids. And as we have hundreds of these amino acids, they're going to form huge long chains, right? So a chain of several hundred amino acids in the correct order, according to the original DNA, is then made. Now this long chain of amino acids, right? It doesn't end here. It's like these genes are super long, and hundreds of these amino acids bonded together. This is what we call a polypeptide. A polypeptide is a chain of all these amino acids that are formed. And lastly, these amino acid chains, the polypeptides, they are folded. So this super long chain of amino acids consisting of hundreds of these amino acids, right? Well, they're going to fold over one another like so. Although it's going to be much bigger than this. It's going to have hundreds of these amino acids. They're going to fold over one another, at last forming a protein. That's it. And the peptide bonds form between the amino acids. So these are the peptide bonds. Peptide bonds. Whereas this long chain of amino acids was a polypeptide. These form peptide bonds between these amino acids, and that's what links these amino acids together, at last forming a protein. And these polypeptide bonds between the amino acids finalize the structure. So let's make an extremely short summary of everything that we have just learned today. So protein synthesis. Sorry for the poor handwriting. Protein synthesis is split into transcription, so which is the shortening of DNA by taking just a single gene that's needed to form a protein, transcription, and translation. So it's when you take the mRNA and you bind it with tRNA and at the ribosome, and you form the amino acids that form proteins. So the transcription is the process of taking DNA and you take a single gene in order to form mRNA, messenger RNA that forms for one particular protein. Translation, really sorry for the bad handwriting. Translation is when you take the mRNA that you've just formed through transcription and you then make proteins with it by binding it with transfer RNAs at the ribosomes. And by the way, here we have what's called an RNA polymerase that kind of acts as a zipper. You guys remember, right? RNA polymerase. It's an enzyme, biological catalyst. So, and you need to know the DNA bases: A, T, C, G, apple tree, car, garage, adenine, thymine, cytosine, and guanine. And in mRNA and tRNA, we have A, U, C, G, because they don't have thymine, they have uracil instead. Uracil and no thymine in mRNA and tRNA. And yeah, that's pretty much it. That's pretty much it. What was wrong with my voice? Just need to know that RNA has no thymine and has uracil instead. And there's complementary strands between DNA and mRNA. So DNA and mRNA, they are forming complementary strands, and they're made with the RNA polymerase. And mRNA and the tRNA also form complementary strands. And the strand from the DNA, right, that we use to copy into mRNA, this strand that we use is called a template strand. And remember, mRNA is single-stranded. And yeah, that is what I believe all you need to know for protein synthesis at GCSE level. Now, I know this was an insanely long video, but I hope at least from watching this video, you're fully able to understand protein synthesis, translation, transcription, and everything. Thanks for watching.